public thesis defense. Matt Hammer is a second-year master's student in the Bioinformatics and Computational Biomedicine program here at OHSU, and he'll be graduating this term upon completion of this presentation and following exam. He started his degree in 2021 after graduating from the University of Vermont with a Bachelor of Science in Biology. His interests are in bioinformatics and computational biology, and it stems from his passion in investigating the multifactorial nature of human health and disease. Along with his coursework and his thesis, Matthew works in my lab as a student worker, and he's been assisting me with some of my research focused on the urinary and gut microbiomes in relation to human health. In the future, he hopes to continue exploring the causative role of commensal microorganisms in human health and disease. And with that, I will turn it over to Matt.
Hello, everyone. My name is Matt, and this is my public oral thesis defense for my project entitled The Evaluation of Functional Inference Tools for the Pediatric Gut Microbiome. Lisa Karstens is my thesis advisor, and the remaining members of my thesis advisory committee are Michael Mooney and Jenny Johnstone. Here's a brief outline of what I'm going to go through in today's presentation. So I'll start with an introduction to the gut microbiome and then address some backgrounds and aims for this specific project, go over the materials and methods that were used, and then talk about results and discuss those results. The gut microbiome refers to the naturally occurring microorganisms that live within the guts of all animals, including humans. The gut microbial community is comprised of bacteria, fungi, viruses, and bacteriophages, as well as archaea, and it contributes to the vital human functions, including metabolism and immunity, among others. The gut microbiome's composition is highly variable between individuals, as well as highly variable within individuals at different times. This makes it very difficult to pinpoint a healthy or normal composition. However, the more diverse the community is, the more stable and resilient that community is to change. And this is important because altered gut microbiome composition has been associated with various different disease states, including metabolic disorders, neuropsychiatric disorders, inflammatory disorders, and certain cancers, among others. Major colonization of the pediatric gut microbiome begins at birth and continues until to develop until the onset of puberty. And there are many factors that impact the colonization of the gut microbiome, and these factors include mode of delivery, breastfeeding status, gestational age at birth, and exposure to antibiotics, among others. These differences in the colonization have widespread effects both during colonization and later in life, making studying the pediatric gut microbiome having a potential to illuminate various aspects related to lifelong human health and disease. Since the colonization begins at birth and the composition continues to develop until the onset of puberty, we see a decreased diversity in the pediatric gut microbiome compared to the adult gut microbiome. And this plot here shows the specific microbial taxa that are differentially expressed in the pediatric and adult gut microbiomes, where orange represents those microbes that are enriched in the pediatric population in this study, and green represents those microbes that are enriched in the adult population from this study. While this is a helpful graphic for visualizing the differences in between the pediatric gut microbiome and the adult gut microbiome, there lacks a consensus for specific differences in microbes present between these two populations, and this is largely due to heterogeneity between individuals and within individuals at different time points, as well as between different populations. It's very difficult to replicate the composition of the gut microbiome in a lab, so we rely on sequencing techniques to identify the composition of the gut microbiome. These sequencing techniques can broadly be broken down into two categories. First being the amplicon-based sequencing, which targets specific regions of the genome known as marker sequences or amplicons. And the second is shotgun-based sequencing, which sequences all desired genetic material in a sample. Of these sequencing techniques, two are most popular in research in the gut microbiome, and this is 16S ribosomal RNA amplicon sequencing, which I'll refer to as 16S RNA for the rest of the presentation, and shotgun metagenomic sequencing, which is also known as whole genome shotgun sequencing, and I'll refer to this as WGS for the rest of the presentation. There are some differences in methodology that are taken by these two sequencing techniques. So 16S RNA sequencing targets the 16S ribosomal RNA subunit gene, and this is found in bacteria and archaea. And this gene is a perfect candidate for marker gene sequencing or amplicon sequencing, as it has conserved regions that are flanked by hypervariable regions, and the conserved regions can be used as targets for primers of amplification, whereas the hypervariable regions can be used to differentiate between specific microbes. This is opposed to WGS, which sequences short fragments from all DNA present in the sample. These differences in methodology reflect key differences in output. So first being 16S RNA captures bacteria and archaea only. And this is because, as I alluded to, this gene is only present in bacteria and archaea, whereas WGS is able to capture all different domains because we are sequencing short fragments from all DNA present. 16S RNA sequencing also has limited taxonomic resolution, and we can really only assign taxonomy reliably down to the genus level, whereas for WGS, we can assign taxonomy to sequences down to the species or even strain level if desired. Third, 16S RNA does not provide functional information, and this is because 16S RNA sequencing is only capable of profiling the taxonomic composition and does not give any information about the metagenome composition. And while all these differences point to the use of WGS for investigating in the gut microbiome, WGS can be very expensive and time-consuming, which can be very prohibitory for certain investigations. To make up for the shortfalls of 16S RNA sequencing, we can use metagenomic inference to infer the metagenome content of a microbial community based on its taxonomic composition. And this relies on the notion that phylogeny is strongly correlated with biomolecular function, and therefore it's possible to predict functional composition of a microbial community's metagenome from its taxonomic composition. There are two common approaches for metagenomic inference, the first being the reference phylogenetic tree-based approach. And in this approach, reference sequences or environmental sequences are placed into a reference phylogenetic tree containing sequences for known microbes with complete genomes. The genomes associated with each sequence are determined based on the placement of the sequence within the reference phylogenetic tree. And metagenomes are constructed by aggregating all genomes associated with sequences identified in a sample. The other common approach is the pangenome construction-based approach. And here, pangenomes for each microbe identified in a sample are generated by combining all genomes from microbes in the reference dataset that have the same microbial taxa. And a metagenome for this is constructed by aggregating all pangenomes associated with each microbe present in a sample. So to summarize what I just went over, and this serves as my motivation for my project, 16S RNA is only capable of profiling taxonomic composition and thus does not provide any information about functional capacity of the environment, whereas WGS is capable of profiling both taxonomic composition and metagenome composition. However, WGS is much more expensive than 16S RNA, and this can be very prohibitory for certain investigations. Functional inference or metagenomic inference offers a cost-effective alternative to WGS for exploratory analyses and can be really helpful for researchers to orient them towards their expected results. There has been some previous benchmarking done on tools for metagenomic inference in the past. However, this previous benchmarking did not include the pediatric gut microbiome, and as I've said, the pediatric gut microbiome is known to vary significantly from that of the adult gut microbiome. and we can't necessarily rely on these results being applicable to this environment. And this led me to propose the following specific aims for my project, the first being to compare popular tools for metagenomic inference, specifically in the context of the pediatric gut microbiome. And using the results that I attained from my first aim, my second aim was to establish a framework for choosing a functional analysis strategy that best suits the needs of different projects as a means for informing researchers who hope to do functional analysis of the pediatric gut microbiome on their options when it comes to functional analysis. And now I'll go over the materials and methods that were utilized in this investigation. This investigation utilized publicly available data that was downloaded from the NCBI SRA database, or sequence read archive. And to be included in this investigation, the data sets needed to meet some specific requirements. This being that the sequences needed to be from pediatric fecal samples. And for each sample, we needed to have paired 16S RNA and WGS data present. And finally, for the 16S RNA sequencing, this needed to target the V4 hypervariable region to ensure consistency across all data sets. We ended up with 3 data sets that met these requirements, the first being the Diabimmune Studies antibiotics cohort, and this contained 39 individuals or subjects from Finland. The second data set came from a study that evaluates differences in gut microbiome development based on gestational age at birth, and we termed this as the gestational age cohort. And this data set contained 41 subjects from New Zealand. The final data set we included here was a subset of samples in the resonance cohort, specifically collected at the second time point. And this contained 41 samples from children in Providence, Rhode Island. Next couple slides go over a demographic breakdown for these specific data sets. We'll start with Diabimmune. The Diabimmune cohort contained 22 males and 17 females with an average age of 1.8 years old. And subjects in this cohort were either assigned to the antibiotics test group or the control test group based on whether or not they received antibiotic intervention during the course of the study. The second data set was the gestational age cohort, and this contained 23 males and 18 females with an average age of 7.9 years old. And subjects in this cohort were either assigned to the very preterm or full term test groups, where very preterm correlates to birth at less than or equal to 32 weeks of gestational age, whereas the full term corresponds to birth during the normal range for gestational age at birth, which is 37 to 41 weeks. Finally, for the resonance cohort, this cohort contained 21 males and 20 females with an average age of 5.2 years old. And this study, the study from which this data set was derived, followed healthy individuals. So there weren't test groups assigned to subjects in this data set when we downloaded the information, but to support different analyses in this investigation that relied on comparisons between test groups, I assigned test groups to each subject based on whether or not they were older or younger than the median age of samples collected at that time, and that median age was 5 years old. So now I'll run through a conceptual model for the methods that I used during this investigation. I started out with paired data, and I extracted the 16S RNA and WGS data. Next, I processed the raw data to ensure that no downstream results were impacted by differences in processing, and I processed these each according to their respective processing pipelines. So for 16S RNA, this consisted of ASV analysis that identifies relevant sequences, followed by taxonomic assignment, where taxonomy was assigned to all the sequences that were identified. For WGS, processing consisted of sequence cleaning, followed by taxonomic assignment. So the result of processing was taxonomic composition for each of these types of data. And to get the metagenome composition for each data type, Metagenomic inference was carried out for 16S RNA, whereas functional assignment was carried out for WGS. Metagenome predictions and metagenomic inference was carried out using 3 different tools, so Paprika, PyCrust2, and PanFP, whereas WGS functional assignment was carried out using Human3. Metagenome profiles here serve as the ground truth or the gold standard for metagenome content in a sample. Here, metagenome predictions and profiles consisted of gene abundance tables where genes were listed on the y-axis with samples represented on the x-axis, and each value in the prediction or in the gene abundance tables reflected the abundance of a specific gene in that specific sample. So metagenome predictions and profiles were generated based on 2 different genome annotation types, EC numbers, which reflect entries in the enzyme commissions database, and KO terms, which represent entries in the KEGG orthologies database. And my reasoning for this was that Paprika's default reference database is based on EC numbers, whereas PanFP's default reference database is based on KO terms. And there's an issue of multi-mapping of KO terms to EC numbers where multiple KO terms will map to the same EC number. And this made mapping between annotation types very difficult. So we decided to just keep these separate because PyCrust2 was able to generate genome predictions or metagenome predictions in terms of multiple different types of genome annotations, including both EC numbers and KO terms. And Human3 as well was able to generate metagenome profiles in terms of multiple different genome annotation types, including KO terms and EC numbers. So once we have the metagenome predictions and metagenome profiles, we were able to carry out benchmarking analysis, which consisted of comparing metagenome predictions to metagenome profiles, and then comparing the results between inference tools. This means that EC predictions were compared to EC profiles, whereas KO predictions were compared to KO profiles, resulting in PyCrust2 being compared to Paprika in terms of EC numbers, and PyCrust2 being compared to PanFP in terms of KO terms. Benchmarking analysis in this investigation can be broken down into 3 different analyses, first being performance analysis, followed by differential abundance agreement analysis, and finally an analysis of the computational resource requirements of each tool. And I'll go into more detail about these in the results section. So let's start with performance analysis. Performance analysis consisted of calculating 6 different metrics, the first being Spearman's correlation, which evaluates the association between predictions and the ground truth, which are metagenome profiles. And here, Spearman's correlation was used as opposed to Pearson's correlation, since we're dealing with a nonlinear monotonic relationship between 2 non-parametric vectors. All but Spearman's correlation are actually classification evaluation metrics that were applied to this investigation. And I'll go into more detail about how these were calculated in the next couple slides. So by converting gene abundance tables in metagenome predictions and profiles to binary, where 0 represents the absence of a gene and 1 represents the presence of a gene, and any gene with an abundance greater than 0 was considered to be present, we can treat metagenomic inference as a classification task. And since we can treat it as a classification task, we can use classification evaluation metrics to evaluate the performance of metagenomic inference. And classification evaluation metrics are calculated based on values in a confusion matrix, which is represented here. And this matrix contains counts for instances of agreement and disagreement between predictions and the ground truth. In this table, true refers to agreement between predictions and the ground truth, whereas false refers to disagreement between predictions and the ground truth. And positive refers to present predictions, whereas negative refers to absent predictions. This means that the true positive count is the number of genes that were identified as being present in both the metagenome predictions and the metagenome profile, whereas the true negative count is the opposite of this, or the number of genes that were identified as being present in both the metagenome predictions and the metagenome profile. On the other hand, the false positive count is the number of genes that were predicted as being present but were identified as absent in the metagenome profile, whereas the false negative count is the opposite of this, or the number of genes that were identified or that were predicted as being absent but were identified as present in the metagenome profile. So here are the equations for calculating each of these classification evaluation metrics. Accuracy is the ratio of correct predictions to all predictions. Precision is the ratio of true positives to predicted positives, and this is most relevant when there is a high cost for having false positives. Sensitivity is the ratio of true positives to actual positives, or the true positive rate. And this is most relevant when concerned with identifying positive outcomes and the cost to a false positive is low. Specificity, on the other hand, is the ratio of true negatives to actual negatives, and this is the true negative rate. And this is most relevant when concerned with accuracy of the negative rate, and there is a high cost for a positive outcome. And finally, F1 score is the harmonic mean of precision and sensitivity, and it takes both false positives and false negatives into account. So this figure shows an overview of the results from performance analysis. And included here is a shuffled null data set, which represents completely random gene abundances generated by chance with no biological significance. And this was generated by shuffling gene abundance counts within samples and then across samples, and this was repeated 10 times. And it's used as a baseline comparison for results due to chance. For Spearman's correlation, we expect the Spearman correlation of the ground truth and the shuffled null data set to be around 0, whereas for accuracy, we would expect it to be around 0.5. From this figure, I won't go into all of the specific plots within this figure, but I just wanted to point out that we observed extremely high sensitivity coupled with extremely poor precision here, and this indicated to us that we might have inflated false positive counts. And this was confirmed by taking a closer look at the confusion matrices, and we found that PANFP had a false positive count of greater than 4,000, which was much greater than any other count, and led us to suspect that spurious gene abundance predictions may be present, and prompted the decision to evaluate different filtering thresholds for gene abundance counts in metagenome profiles or predictions. This table shows an overview of the results from comparing the performance analysis at different filtering thresholds. Here we can see that we observed a significant increase in Spearman's correlation, accuracy, precision, and specificity for both Paprika and PANFP, whereas we observed a decrease in sensitivity, but this was expected since filtering decreases the number of positive predictions and sensitivity measures the ratio of positive predictions. We also see that there was no significant change in performance for PyCrust2. We confirmed these trends by taking a look at the false positive counts after filtering, and we noticed that the false positive counts decreased substantially after filtering for both Paprika and PANFP. So Paprika decreased by around 300, whereas PANFP decreased by around 1,700. And this decrease was reflected in an increase in the true negative counts for both Paprika and PANFP, where essentially all of the occurrences of false positives were transferred to true negatives. And these results indicate that gene abundance count filtering effectively improved performance by removing potentially spurious results. So now this figure shows an overview of the performance results for metagenome profiles with gene abundance counts filtered at a threshold of 1. And each of these metrics that are represented here describes a different aspect of performance. And while they're all helpful and informative, there are certain metrics that are more relevant for this investigation. The first being Spearman's, as this was used to confirm the efficacy of the shuffled null data set. Accuracy is also important because we're concerned with correctly identifying present and absent genes. Sensitivity is important because we're concerned with identifying all potentially present genes and do not necessarily care about false positives, as it's more important to identify potentially present genes than misidentifying a gene as being present. However, F1 takes into account the false positive rate, and while we're concerned with identifying all potentially present genes and don't necessarily care about false positives, we don't want our results to be too heavily skewed by false positives, so F1 takes into account the false positive rate, and that's why it's important for this investigation. So we'll first take a look at the results from Spearman's correlation analysis, and this indicates the association between predictions and the ground truth. And as I mentioned earlier, this mainly serves as a confirmation that shuffling for the shuffled null data set was successful, as we would expect to see very little correlation with the ground truth for a completely random data set, and that's exactly what we see. So here and here and here and here, we can see that the Spearman's correlation between the shuffled null data sets and the ground truth was essentially 0 as expected, and it indicates that these data sets serve their intended purpose. We can also see from this figure that PyCrust2 exhibits significantly greater correlation with the ground truth than other metagenomic inference tools. And all tools exhibit significantly greater correlation with the ground truth than the shuffled null data set, which indicates that associations between each metagenomic inference tool and the ground truth are biologically relevant. Next, we can take a look at accuracy analysis. And we observed some varying trends in accuracy across the different cohorts. So we'll start by looking at the diabimmune cohort. And in this cohort, we see that paprika exhibits significantly greater accuracy than PyCrust2 in terms of EC numbers. And since both tools exhibited significantly greater accuracy than the shuffled null data set, we can assume that this finding is biologically relevant. However, for KO terms, both tools exhibit significantly less accuracy than the shuffled null data set in terms of KO terms. And this indicates that KO term predictions are less accurate than by chance in this specific cohort. Moving on to the gestational age cohort, we see again that paprika exhibits significantly greater accuracy than PyCrust2 in terms of EC numbers, and both tools exhibit significantly greater accuracy than the shuffled null data set. So we can assume that this finding is biologically relevant. As for KO terms, we see that PanFP exhibits significantly greater accuracy than both PyCrust2 and the shuffled null data set in terms of KO terms. And we see that PyCrust2 actually performs worse than the shuffled null data set in terms of accuracy for KO terms. And this indicates that, again, for this cohort, that PyCrust2 predictions are less accurate than by chance. Finally, we can take a look at the resonance cohort, and in this cohort, we see that all predictions are better than have significantly greater accuracy than the shuffled null data set, which tells us that these findings are biologically relevant. And we see that PyCrust2 exhibits significantly greater accuracy than paprika in terms of EC numbers, whereas PanFP exhibits significantly greater accuracy than PyCrust2 in terms of KO terms. We can interpret these results as being that accuracy for each tool varies depending on the cohort being analyzed. However, we do see some trends where paprika exhibits significantly greater accuracy than PyCrust2 in terms of EC numbers in 2/3 of the cohorts, whereas PanFP exhibits significantly greater accuracy than PyCrust2 in all of the cohorts, except that in the first cohort, both exhibited significantly less than the shuffled null data set, so we can't really use that in our findings. So now we'll take a look at the sensitivity analysis, and this measures the true positive rate. And here we observe the same trends across all the cohorts that we investigated. So for all cohorts, PyCrust2 exhibits significantly greater sensitivity for each genome annotation type. And since each tool exhibits significantly greater sensitivity than the shuffled null data set, we can assume that these findings are biologically relevant. We can interpret these results as PyCrust2 predicting more genes as being present compared to other tools. However, since PyCrust-2 is unaffected by gene abundance count filtering, this could be an artifact of inflated false positive counts. And this actually appears to be the case, especially for KO terms, where the false positive count greatly exceeded all other counts. So while we're concerned with identifying all potentially present genes, we don't want the majority of those genes to be misidentified, which is the case here. So PyCrust-2's greater sensitivity does not necessarily correlate to improved performance for these cohorts. So finally, we can take a look at the F1 score, which is the harmonic mean of precision and sensitivity. And as I mentioned earlier, it takes both the false negative and false positive counts into consideration. We observed some trends that were consistent across all cohorts, and specifically for KO terms, where PAN-FP exhibits significantly greater accuracy, or F1, than PyCrust-2 in terms of KO terms for all of the cohorts. Additionally, all predictions were significantly greater than the shuffled null dataset for each genome annotation type, indicating that all of these findings are biologically relevant. As for EC numbers, we see that in the diabimune cohort, there was no significant difference between Paprika and PyCrust-2 in terms of EC numbers, whereas in both the gestational age cohort and the resonance cohort, we see that PyCrust-2 exhibits significantly greater F1 than Paprika in terms of EC numbers. These results can be interpreted as PAN-FP accurately predicts more genes as being present than PyCrust-2 in terms of KO terms, and PyCrust-2 accurately predicts more genes as being present than Paprika in terms of EC numbers. So next, we can take a look at the results from differential abundance agreement analysis, and this measures the overlap of, or the agreement of, differentially abundant genes identified in both the metagenome profile and the metagenome predictions. And I used a chi-squared test to test for the significance of this agreement. This analysis sheds light on the predictive power of metagenomic inference tools and specifically evaluates whether results from differential abundance analysis based on WGS can be replicated using metagenomic inference. So these are the results from differential abundance agreement analysis. The top tables represent the counts of differentially abundant genes that were identified by each tool, whereas the bottom represents the agreement analysis. So as we can see in both the diabimune cohort and the gestational age cohort, we didn't find any differentially abundant genes that were produced by any of the predictions or the metagenome profiles. And while this is a slightly striking result, it's not necessarily that surprising based on the results of our exploratory data analysis, where we compared the taxonomic composition between test groups in each of these cohorts. And in this EDA, we did not identify any significant difference in taxonomic composition between the test groups in the diabimune cohort or in the gestational age cohort. However, we did identify a significant difference in taxonomic composition in the resonance cohort, and this was reflected in the fact that we did identify differentially abundant genes also. Here, we identified 14 genes as being significantly differentially abundant by WGS using EC number genome annotations. We also identified 119 genes as being differentially abundant using PyCrust-2 in terms of EC numbers, and 394 genes as being significantly differentially abundant for Paprika. Of these genes that were identified as being significantly differentially abundant, 1 from PyCrust overlapped with WGS, whereas 4 from Paprika overlapped with WGS. And this resulted in a kappa value of essentially 0, which indicates that agreement was no better than by chance for this. And finally, now we can take a look at the computational resource requirements analysis, and this just compared memory requirements in terms of RAM and storage requirements in terms of disk space. So here we can see that WGS processing using Human 3 required far more computational resources than any of the metagenomic inference tools. Within the metagenomic inference tools, PyCrust-2 had the most computational resource requirements, followed by Paprika, and then finally by PanFP. So now we can discuss what these results actually mean. First, I just wanted to introduce the functional analysis decision framework that was generated based on the results of this investigation. And this was generated as a means of helping investigators choose a functional analysis strategy that best suits the needs for their specific project. So when we decide that we want to do functional analysis, our first decision that we need to make is what is our desired functional output. If gene content is our desired functional output, then we have a choice between two different approaches. The first being metagenomics, which is done using WGS, and the second being metagenomic inference. The deciding factor here is the cost and the confidence of attaining results. So for metagenomics, we have high confidence in our results, but also high cost in attaining those results. Whereas if we're more concerned with the cost of attaining results, and we're not necessarily as concerned about the confidence in the results as we would be in an exploratory analysis that's going to be validated anyway, then we can use metagenomic inference. And deciding between the metagenomic inference tools really comes down to the genome annotation type that's being used. So if we want to use KO terms, then we can use PanFP. However, we identified that KO terms are subject to spurious results. If we want EC numbers as our genome annotation type, then Paprika can be used. Additionally, if we want multiple genome annotation choices, then we should use PyCrust2, and we can use PyCrust2 to generate KO term predictions and EC number predictions, as I mentioned earlier. And another factor that plays into this decision is the computational resource requirements. So we observe increasing requirements across these three. So PanFP requires the least amounts of computational resources, followed by Paprika, and then PyCrust2 requires the most. If gene expression content is our desired functional output, then we really only have one option, and that's to carry out metatranscriptomics, and this is using RNA-seq. The same goes for protein content. We really only have one option, and that's to carry out metaproteomics. However, we do have a choice between what tools we want to use to carry out metaproteomics. So we can either use normal mass spec or a newer version of mass spec, liquid chromatography mass spec, and this just results in higher confidence results. And finally, if metabolite content is our desired functional output, we again have the choice between metabolomics or metabolic model inference. And this, again, the deciding factor here is cost and confidence. So if we're really concerned with having high confidence in our results, then we want to carry out metabolomics, and this is done using mass spec or nuclear magnetic resonance spectroscopy. Whereas if we're not necessarily concerned with the confidence in our results, but we really are concerned with the cost of attaining those results, like we would be in an exploratory analysis, then we can carry out metabolic model inference. And there's one tool available to do this right now, and that's a tool called MyCom. So as I've kind of alluded to earlier, metagenomic inference has some limitations. The first being that it does not provide evidence. All the findings that we get from metagenomic inference do need to be validated to draw relevant conclusions from it. But this doesn't discount the use of metagenomic inference, as it can be really helpful for orienting researchers towards expected results. And this can be helpful for grant writing or hypothesis forming or other activities like that. Additionally, metagenomic inference is susceptible to errors resulting from horizontal gene transfer. And this is because we're relying on the reference databases to predict the metagenome content or the genomes of the microbes that are present in the gut. And we're not actually measuring what's going on within the gut. And this means that if there's horizontal gene transfer between microbes that's happening within the gut of a specific individual, and that's not reflected in the reference database, then we're not going to be able to capture that horizontal gene transfer. Additionally, metagenomic inference relies on reference databases, and this means that we're limited to those microbes that are represented in these reference databases, and this can cause issues if we're investigating environments that are not well reflected in these reference databases, like the pediatric gut microbiome or other microbial environments that aren't necessarily as studied as the adult gut microbiome, for example. And finally, metagenomic inference does not predict metagenome contribution from all domains, and this is because it relies on 16S RNA sequencing, which is only capable of capturing bacteria and archaea. There are also some specific limitations to my analysis plan. The first being that I relied on default reference databases, and there were some differences between the default reference databases that were used by each of these tools. And also for each of these tools, the reference databases that were provided by default, there are more current versions available for each of these. Additionally, I relied on publicly available data, which resulted in gaps in age, as well as gaps in representation of different diets and gaps in representation of different ethnicities. And all of these factors are known to have a significant effect on the composition of the gut microbiome. Additionally, there were discrepancies between genome annotation types, as I mentioned earlier. So PanFP produced metagenome predictions in terms of KO terms, whereas Paprika produced those predictions in terms of EC numbers, making it impossible to directly compare Paprika to PanFP. However, we used PyCrust as a sort of proxy for comparison between the two. And finally, this being a master's thesis, I was subject to time and computational constraints. Given more time and expanded data sets could have been utilized, which could have resulted in more conclusive results, as well as some more nuanced investigations. So we can draw some, we definitely can draw some conclusions from this investigation. The first being that metagenomic inference does serve as a cost-effective alternative to metagenome profiling for exploratory analyses in the pediatric gut microbiome. And there's an asterisk here because, as I mentioned before, results from exploratory analysis based on metagenomic inference do need to be validated using other methods before conclusions can be drawn. Additionally, we observed high variability in the performance of metagenomic inference tools. Specifically, results from performance analysis varied depending on the data set being analyzed, as well as the genome annotation type used and the metric being compared. We also see that KO term genome predictions were subject to inflated false positive counts. And we also observed disagreement with previous benchmarking, specifically benchmarking done by the PyCrust 2 developers, in which they reported that PyCrust 2 performs better than PanFP. They didn't include Paprika in their investigation, but this disagreement is likely due to the fact that we're surveying a different environment, as well as the fact that they didn't carry out any gene abundance count filtering in the previous benchmarking. And in our investigation, by filtering gene abundance counts at a threshold of 1, we effectively removed spurious results and deflated the false positive count for PanFP. So based on these conclusions, I came up with 3 recommendations for metagenomic inference. So if PyCrust 2 is being used, then EC number genome annotations should be used rather than KO term genome annotations, and this is because of the issue with false positives and spurious results. Additionally, if using PanFP, then gene abundance count filtering at a threshold of 1 should be considered. And finally, if KO term genome annotations are desired, then PanFP should be used rather than PyCrust 2 based on the results from performance analysis. All right, so that's all I had. This is my references. And then are there any questions? And also, I'm going to transfer over to the microphone in the actual room, so you might hear some static going on. I couldn't hear if anyone said anything, so if you had a question, please repeat it.
So thank you, Matt. If anyone has any questions that are online, please let me know by raising your hand, or you can just unmute yourself and start talking. So really nice job. Does anybody that's in person here have a question? And the committee members, if you have more detailed questions, we can get into that after in the private one. But yeah, if you want to go ahead, Jenny.
So thank you for a great presentation.
Thank you.
And I noticed a lot of, you took a lot of input, so that's great. So in your analysis plan, you noted limitations, and one of the things you said is, limited like ethnicity or maybe accounting for that. Can you just say a little bit about gut microbiome differences from an ethnicity standpoint?
So there have been, like I said before, it's really hard to pinpoint specific normal or healthy compositions for different populations. However, there is a lot of heterogeneity between different populations, and that different ethnicities is not excluded from that. So there are specific microbes that are definitely more present in specific ethnicities, and also based on ethnicity is kind of related to diet in certain times, and diet really has a huge impact on what microbes are present in the microbiome. However, there is some agreement in the genes that are produced by these microbes. So we do observe pretty similar functionality, but there are differences in specific microbes.
Do you have any questions?
I know you mentioned that the data you were using, they were separated into groups that were being compared.
Mm-hmm.
Can you talk a little bit about the way that you grouped that one data set that was not grouped, and how that all worked?
Yeah, definitely. So because the gut microbiome the colonization begins at birth and continues until the onset of puberty, I made the assumption that there would probably be differences in the taxonomic composition at different time points within that range that was represented by that study, and we actually found that to be true. So we used the median to just essentially split the whole data set in half, and since we used age, we would expect to see at least a little bit of a difference between the two test groups, but between older and younger, and that's exactly what we saw. So that's why I decided to use median age as my kind of threshold.
Thank you. Now, I have a question, because when you were showing a lot of your accuracy and sensitivity results, a lot of the results with the KO terms, I believe it was, they showed the same trend in 2 of your cohorts, but not in 1. And the one that was different was always diabamune. Is there something specific about that cohort that makes the predictions a little less accurate? Or do you have any thoughts on why that tended to be the case?
So I guess there's a couple reasons why that could be the case. So the diabamune cohort, the subjects in the diabamune cohort were by far the youngest subjects that were identified. So there could be, because it followed children from age 0 to 3. So it isn't necessarily that surprising that there wouldn't be that much of a change within the composition between the ages of 0 and 3, just because it's a pretty narrow time frame. But there's also other things that can contribute to it. The fact that it's all individuals from Finland, which is a kind of special case. The Scandinavian countries are pretty known to be almost have like a founder effect type deal, where they show different results than other types of populations. And then also the antibiotic intervention could have had some impact on what was happening.
Thank you. Are there any questions from everyone that's online, or anyone that's online? I see that Karen has a question. Can you unmute and ask it?
So nice presentation, Matt. And I guess I would just ask you to think forward, if you were to extend this research, you put forward some recommendations, but what direction would you go if you were going to continue this research?
So first, I would include way more data sets. I would include expanded data sets. As I mentioned, I did take a subset of some of these data sets, and this was just to accommodate the computational and time constraints that I was under. I would definitely include expanded data sets, which could result in more conclusive results. I would also do some different analyses. I would probably use a more stringent or nuanced approach for differential abundance analysis because I used a pretty basic approach for this, and I think that I could potentially see better results, more conclusive results using a type of differential abundance analysis that maybe takes prior knowledge into account, like a Bayesian approach, or one that adjusts for or accounts for covariates, or even in the microbiome, we deal with very sparse data, so that could have a big effect on it too, so filtering.
That's very helpful. And maybe I missed it. You may have shown it. Did you show like area under curves when you were talking about accuracy?
No, I didn't include the area under the curve, but that is kind of represented in the F1 score. But that's another thing that I would do. I would I would include some expanded performance metrics and do a more deep dive into the actual differences in computational resource requirements. There are plenty of other different types of analyses that I could have done. Sure. Additionally, I could try to, one thing that I have recently been considering is the fact that PyCrust2 was not responsive to the filtering, whereas, but we still saw a lot of spurious results for the KO terms. So evaluating more stringent thresholds for filtering may result in better results for PyCrust2 in terms of KO terms. But in general, I would probably lean away from using KO terms. One, because it's a proprietary database now. It was at one point open source, but now it's proprietary, which is one of the reasons why the databases haven't been updated in a while as well. Additionally, I would also probably include custom databases because I could probably get better results.
Thank you.
So are there any more questions from the folks online? I do not see any. Anyone here? Right?
Mm-hmm.
Did you see anything like diversity?
Not specifically. Besides what I alluded to earlier, the fact that we didn't really see any taxonomic differences or taxonomic composition differences between the cohorts, that definitely could result in differences. And we did see that I believe the diapamine and the gestational age cohort were more similar than the resonance cohort. And that could be a result from the fact that there's not really that big of a difference between taxonomic composition, but it also could be a result of the fact that we could be slightly underpowered for this effect size with this sample size that we're using.
And then, yeah, I guess that was my next question, was about the whole bucket.
Mm-hmm.
Like, what is your interpretation of the results first? Do you have high confidence that those differences that we could see are real?
I would definitely need to look back and do some more analysis. I do think that we did observe what we kind of set out to see, which was, can we replicate the results from WGS in with metagenomic inference? However, I definitely think that using a more nuanced approach for differential abundance probably would have resulted in more conclusive results, definitely.
Do you know what some common critiques of differential abundance analysis are?
Yeah. So as I mentioned earlier, there's the sparsity is an issue. So the fact that in metagenome profiles, we see a lot of genes as being, and even in just normal relative abundance of specific microbes, if we're comparing the differential abundance of that, we see very sparse data where we'll see lots of microbes having zero abundance, and these can really skew the, if specifically for our investigation, we were comparing, I used a Wilcoxon rank sum test to just compare the distributions, and these were heavily skewed by the fact that there were a lot of zeros present.
Great, thank you. If there's no more questions from the online audience, we will end the meeting, and then Matt will stay for meeting with his committee members. Thank you.
Thanks, everyone.